Thank you, Steffi, and thank you to all of you because you've made it to the end of a very long day, and you've come to a talk called Urban Metagenomics, and the likelihood that you know what that means is roughly zero. So that's why I'm going to do this talk, is to sort of talk about something that as a field didn't really exist even, say, 2-3 years ago, and is really just coming into being, and what it will mean for all of us is quite unclear and quite exciting. I'm going to start by talking about this piece, an artwork in Bristol. This is an artwork called Seeds of Change by the artist Maria Teresa Alves, and it's what she calls a ballast garden. And ballast is that it's a funny word, that refers to the kind of the basement of the ship, the part that basically keeps the ship from toppling over. And if your cargo was light during the age of exploration and colonization and so on, you'd have to pack that full of whatever you could find to stop the ship from tipping over. And so that's what the age of the explorers, et cetera, would do. They would just take whatever was handy and they would just throw that into the bottom of the ship. And typically, those were stones and rocks from wherever they happened to be leaving from. So a ship might look like this. That whole bottom layer is just stones from Spain on their way to Florida, which is where they get excavated. So a shipwreck in Florida, all the wood has rotted away, all the cloth has rotted away, but these stones are actually from Spain. And so this is the interesting part, is that these sailors, the explorers, the colonizers, whatever you want to call them, when they were packing those stones into the bottom of the ship, they were packing what were essentially Spanish rocks without thinking about the fact they were also bringing Spanish dirt, and that in the Spanish dirt were Spanish seeds, and that from those Spanish seeds, a wide variety of new flora actually came to the United States. Nobody meant to bring it there, nobody set out to bring it there, but nobody could stop it from coming. And this happened all over the world, basically through the 19th century, and it led to a variety of invasive species and propagation that was sort of just this weird byproduct of colonization. So Maria Teresa Alves did this beautiful work called Ballast Garden to sort of talk about that. So this is in Bristol, England, and she basically planted a garden just with the flora that arrived by accident from these ships, things that are now native to England, but really arrived in this weird way. So that includes marigolds from the Mediterranean, and Aruca sativa, and Ficus carica. And these plants, they arrived and they were kind of like these, quiet migrants, right? Nobody really noticed that they were there along for the ride. And the question that I don't know Maria Teresa Alves, but I think the question that's provoked by the work is, sort of who exactly colonized whom, right? That the process of colonization worked in one direction, sort of socially and economically and politically, and worked in just the opposite direction in terms of nature and biology. Because no captain was ever aware of how much of their cargo was actually alive. And I mention all this because, of course, neither are we. And we've heard this referred to actually in the last 3, maybe 4 talks, the gut biome, what was always said to be the 90% of your DNA that isn't you, and maybe it turns out to be more like 50%. It may turn out to be that only 50% of the DNA in your body isn't actually human. It's somewhere around 2 kilos of you that is not human at all. It's basically a bunch of between 500 and 1,000 in a healthy individual organisms at the scale of trillions that have their own agendas. All of them are broadly beneficial to keeping us alive. Not all of them, and when they go out of balance, that's when things start to go awry in human health. And so that leads to things like obesity and disease. We've heard this a couple times now tonight. Obesity and disease, and even fundamental human behavior. And here's, by the way, where it gets kind of weird to me, which is that we're becoming increasingly aware of the relationship between specific bacteria in the gut, and things like schizophrenia, things like obsessive compulsive disorder, which is related to streptococcus, which The theory on this is that Streptococcus produces a shift in the basal ganglia of the brain that gets you to wash your hands over and over and over again to prevent healthy new bacteria from battling against the Streptococcus. So depression is well linked at this point to what's happening in the gut biome, and even the likelihood that you will crash your car or have unprotected sex can be traced back to specific bacteria that live in roughly 33% of humans on Earth. And so, you know, the question, if you really start to like delve deep into this from a sort of genomic and genetic level, is that, you know, in 1999 or so, we sequenced the genome, and that was supposed to unlock all the mysteries of human existence, and it turns out it sort of doesn't. It's really just the very beginning, because so little of us, relatively speaking, is actually human, is actually run by the DNA that we're running. And that if you wanted to go ahead and sequence what's important about, for example, this woman here, yes, you could grab her genome, and you could do that for whatever it is now these days, $200, maybe $1,000. But what about everything in there? What about the 500 to 1,000 that's in there? And there are actually startups. uBiome is a really good startup out of San Francisco that will sequence the biome in there. But the question is, those 500 to 1,000, where did they come from? Right? These other species that keep us alive, where did those come from? And the answer is basically, you know, here and here. And how do we sequence that? And that is the ability to even think about how to sequence that big black mass that affects everything about us is what's really new. And it has to do with a couple different things. It's one of the unexpected bounties of cheap genomics. You know, we think about cheap genomics, okay, now that means that everybody can get their DNA tested for cheap, but it also means that all things in the world can be sequenced cheaply. And so what metagenomics refers to is basically sequencing all kingdoms of life and their DNA. In other words, to take like this glass of water and say like, I don't even know what's in there. I'm not trying to target something in particular, but just show me everything that's in there at this sort of meta level. What is the interaction between all the kingdoms of life as I can detect that genetically, and actually to collect that from the environment. That's this broadly new field, metagenomics. And it's important because we learn that the gut biome, which is, again, this thing that keeps us alive, it's at least half of us, it's basically an exchange with three things in the world. It comes from your mother when you're born. It comes from the food that you eat over your lifetime. But really, it comes from everything that you touch and everything you breathe and everything that you're exposed to, even obviously right now. And of all the ways that, in the ways that this type of life starts to get compressed in terms of the human experience of it, it really is unique in the density and diversity of these weird biological superstructures that we call cities. That there's something about the density of this invisible strata of life, biologically speaking, that is in some sort of dialogue with human existence, behavior, and health. And we're just beginning to learn about that because we live among all this sort of unaware of the live freight of the cities. I come to all this as a designer. I'm not a scientist. I work with some really badass scientists, but I know very little about it. Actually, I learned from them. This was inspired specifically by the work of good friends of mine. I have a company called Audica, who did something that what they called immaterials, where they were showing, okay, RFID, the little chip that's on everything that you own these days, you know, can we begin to understand what the invisible field around that is? And then they extended that out to an urban scale to start to see, can you get visibility of Wi-Fi in the city? You know, does it have a life that we could begin to understand? And this is really beautiful, beautiful work. But the truth is that invisible radio waves are sort of, you know, they're nothing, you know, compared to the invisible living kingdom that surrounds us all the time. And the first time that I saw any evidence of people who were thinking about this was just, maybe it was 2 years ago. It's not more than that. And it was a not super well-known paper of some people in Hong Kong who sequenced at a metagenomic level everything that was happening in the subways in Hong Kong to start to understand Is there a difference from stop to stop? Is there a difference between times of day and so on and so forth? Not in the human part of it, but in this other part of it. And they published a paper that I think is sort of the first and has led to just the beginning of a bunch of interesting work in this field. So I'm going to talk about 4 different labs that are working on this. Mine is one of them, but I think in a way the least interesting, certainly the least scientifically rigorous. The first one is Chris Mason at Weill Cornell in the Chris Mason lab, and they have what they call the PathoMap project, and they are sequencing the metagenome, the metagenomic profile of the New York City subway. And that meant actually physically swabbing, like you see there, it's not a sexy job, actually swabbing the 468 subway stations of New York City, and then just taking whatever it is that you find there and just sequencing it and seeing what gives. And it's, this is early stages on all of this, right? So nobody, it's sort of like somebody just built a microscope and now they're like, but what is that actually? And you can start to see like, so Enterococcus, is extremely popular on the Upper West Side, whereas Staphylococcus is extremely popular in Coney Island, which is not probably an excellent thing. But here it gets extremely interesting. So this is one that I can't even pronounce, but it refers to the microbiological environment of that particular subway station, which had been flooded by Hurricane Sandy and is essentially, at this invisible microbiological level, a marine environment, right? You don't know it, you don't necessarily smell it or feel it, but around you, it is it has more to do with the sea than certainly any other station on the subway, and maybe than it should. Of the many, many interesting things that you discover when you start to really look at the world like this, this was Chris Mason's work, is that so 48% of what they find has never been classified, has no names. This is an entire strata of life on Earth that we are, in real terms, just coming into contact with. So to me, this idea that like, yeah, we're going to get to Mars, but we don't know anything about what's happening in the subways is weird. This was a very beautiful thing that they found. That one station there indicates high prevalence of specific bacteria, which is produced in the fermentation process of kimchi and sauerkraut. So what you see there is basically Koreatown in New York City. You see that this is where a lot of people eat kimchi. And when they eat it, that moves through their system. Basically, they shed it off through skin cells or through their breath or their spit or whatever it is. And it means that those subways have a certain form of life that is unique to the long effects of the people who've been there, and that the health benefits of the kimchi that they have eaten may well extend to the people who come into contact with them. Maybe, I don't know, supposition. So that's Chris Mason's lab. This is Eric Alm's lab at MIT, which is, it's a different part of MIT than what I'm in, and this is actually in conjunction with Carlo Ratti's lab, the Sensible City lab, and they've been working on something called Underworlds, the Underworlds project, which is urban metagenomic profiles. This is their slide. What do they say? What is in our collective gut as a city? And if you thought that swabbing the subways was a really unsexy job, this is what they do at the Alm lab. That's the open sewer there. And these are the sewer robots, Mario and Luigi, that they've built that are doing metagenomic profiles of the sewers of Cambridge. And we are starting to learn. This is like really, really early stages. In fact, all they've mapped publicly are the viruses. There's still a lot left to do. We're beginning to learn, like even just from an epidemiological perspective, how this invisible layer of life upon which we are dependent and affected is moving around, and how it surfaces and what it does. The third lab I'll show you is Jessica Green's lab at the University of Oregon. She has a lab called Biology in the Built Environment Center, which does what it says it does. It studies biology in the built environment. This is her work here. She's basically studying how the physical environment, spaces like these, what the microbiological world in these looks like and how it's affected by its design, by its use, by its age, and so on and so forth. And there's a couple of things that they have discovered just in early work on this. So number one, they didn't discover this, but it's just true and pathetic, which is that on average, in general, in the Western world, we spend 90% of our lives indoors. So all this talk about the microbiological layer of the city is really, in a way, 90% of it is the microbiological layer of spaces exactly like this one. A lot of what's in the built environment, perhaps unsurprisingly, but is now confirmed, is human bio emission, which is to say, if you were to sample what's going on in here, it wouldn't be a lot about soil or trees or what's going on. It would basically be you. It would be basically be the clouds that you and I are all leaving behind based on our lives and what's happening essentially in our gut. That fingerprint that we leave behind is unique, if maybe a bit cloudy. That's one of the things that Jessica Green's lab is studying, is the idea that you can be fingerprinted, that any one of you, when you get up from your chair today, if I sampled that and I had sampled something else that you had sat on at another time, could say, yes, that's you, because that is you. It's not the human part of you, but it's you nonetheless. One of the findings that she did that I think is just sort of one of the most remarkable and really speaks to the German fetish for Frischluftkultur, turns out to have a strong scientific basis, perhaps unsurprisingly. But this is the science behind it, and what you see here is that in spaces like this one that are mechanically vented, right, where the heating, ventilation, air conditioning is basically conditioning the air all around us, you see two things. You see extremely low diversity in this microbiological level around us, and you see a very high likelihood of potential pathogens being within that. When you go outside, you have far greater diversity. Again, none of this is surprising, it's just that we can measure it now, and we can see it, and we can start to understand how to build for it. When you go outside, far greater diversity and far lower likelihood of potential pathogens being in there, right? So the likelihood that you're going to get the flu from being in this room is much higher than by walking in the streets of Munich. There's a reason for that. There's science behind it. Anyway, from my perspective, what we're working on at the Media Lab, I come to this from a design perspective, and it comes from basically just wanting to begin to build an imagination and a conversation around what's happening. It's inspired in part by this, which is in my apartment in New York. This is a work from quite a while ago by Takashi Murakami for the exhibition Happiness at the Mori Art Museum. And it's really beautiful, and it's this idea that's Roppongi Tower there of sort of happiness sort of coming out of that. And I look at that basically more or less every day and have always wondered, what is that actually? Like what is that happiness that Murakami renders there? And then the question is, other than sort of getting hundreds of people to swab the subways for you, or putting robots into the sewers, or building synthetic environments that you can measure carefully, how else could we get a good reading of the invisible kingdom around us? And then we remembered the story of the red honey of Brooklyn. I don't know how many people are familiar with this. Urban beekeeping is a really big thing if you have a beard and tattoos. It just correlates strangely. There were some beekeepers in Brooklyn who went to get their honey and were justifiably alarmed to find that it was bright red. And the question is, why? How? How could, and is it healthy? Is it dangerous? And so what they had to do was sort of become bee detectives and figure out what the bees had gotten into. The answer is super obvious when it's the maraschino cherry factory that was nearby. That they were getting drunk on the syrup from the cherry factory. The Environmental Protection Agency moved in because that shouldn't happen. And when the Environmental Protection Agency moved in, they also found that, by the way, it also turned out to be the biggest illegal marijuana growing farm in the history of New York City that was discovered by the bees. So, if we learn something about Brooklyn, Could we learn something from the bees about Tokyo? We're working with the Mori Building Corporation. So if bees can sort of tell us about maraschino cherry factories, what else could they tell us? And could we start to actually use beehives as metagenomic sensors for the urban environment? And so we've started this. This is work in progress, so I don't have a lot of sexy results to tell you. I can just tell you what we're doing. We're working with hives in Brooklyn and in Tokyo, and just as of next week, 15 hives in Australia, where bee season is starting. And we work with what beekeepers like to call bee debris. Bee debris is a good euphemism for bee poop. It's all the parts of the hive that the bees don't really like or want. It's not the honey, it's not the good stuff. It's everything that they leave behind. And everything that they leave behind is basically everything that they've come into contact with in the 2-mile radius around their hive. So we sample that, we purify it and sequence it. That's what this looks like, and this is sort of some of the process of doing that. Then we bring that up to the Chris Mason lab, Elizabeth Hanaf, and it goes in for metagenomic sequencing. And by the way, part of that is just the cheap genome, but also the computational power to try to figure out like all the DNA of all these things and you don't even know what you're looking for is actually immense, right? I mean, like as data projects go, this is madness, and it takes a long time, and there's a lot of difficult work that goes into it, and that's underway right now. And in the meantime, the question is how to adapt existing beehive infrastructure to optimize for getting what we want out of it, which is the bee poop. So this is inspired by old photographic plates, you know, where they had to sort of protect them from the light when they were in the camera. So this is a tray that goes underneath the hive. That when it is inserted in, will actually sort of open up to allow all that to propagate in there. And then when you pull it out, it's sealed. We can send that to the lab and figure out what's going on. So we're designing that infrastructure for the interfaces between standard and non-standard, because there's this interesting thing about beekeeping that it's weirdly standardized, which may not really be good for bees, but it's a fact, and we're working with the infrastructure that exists. But we're also thinking about how to start building new hives for bees that are optimized, have nothing, like we don't care about the honey, and we don't care about all these other things that people are usually trying to get from the bees. We just want this thin layer at the bottom. So we've got the first layer of metagenomic data back. It looks like this, it looks like this, it looks like this. And on our way to figuring this out, there's sort of 4 hypotheses that are borne out. One is that there is a sort of microbial fingerprint that's common to all beehives, but you go from neighborhood to neighborhood in an urban setting, and they do differ. They can differ quite a bit, and a beehive that's within 2 miles of another beehive will look very similar to that beehive in a way that one that's 3 miles away will look very, very different. And that the idea that we can start to use these as sensors seems to be quite real. Most of what we find is from soil and freshwater, which is good. It means we're not getting sort of false reads. But we basically need a lot more hives before we can start doing, getting really meaningful data out of it. So our next steps are getting more and better data out of all this, more hives, eventually to sort of discover how is it that we are interacting with this invisible layer the same way as with our gut. Right? Like in the same way that we're just beginning to understand the relationship between what happens here or here and this, how do we understand the relationship between this and this? But from my part, this is not really an exercise in gathering data, but actually to sort of build the imagination, and to build the imagination of something that's totally real, and to get us to the place to understand that, like us, and like the ships that carry us here, that cities have cargo that might secretly be the captain. So thanks very much. Thanks for listening.